मैं जो है वो फूड टेस्ट को आपको जल्दी से रिवाइज करवा देता हूँ so that it serves as a good revision for us. Okay, if you can just give me a second. Animal nutrition food tests. Okay. So, okay, so that's a table I have. Very quickly, we are going to discuss the reagents, we are going to discuss the method, and we're going to discuss the positive and the negative results. All right, whatever the case is, just ignore it says your internet connection is stable. When internet runs out of its mind and says the signals are not good when they're good. Acha ji, so bacho, there are four substances that we need to know and understand that are to be tested: starch, fats, proteins, and reducing sugars. इसमें जो आपने बात याद रखनी है वो ये है that two of them are from the same class of food: starch and reducing sugars. Starch and reducing sugars belong to carbohydrates, and then fats, of course, to fats, and the proteins, of course, to proteins. We need to know the names and colors of the reagents of all of these four substances to be tested. Starch के लिए हम use करते हैं iodine solution, which is brown in color. Fats के लिए we use ethanol, which is colorless. Proteins, Biuret solution, again it's blue in color, and Benedict solution, of course, is again blue in color. So Benedict, Biuret, both of them have blue color. Ethanol is colorless, and iodine is a brown orangish solution. Okay. Out of all of the three tests, it's only the reducing sugar test जहाँ पे heating required होती है. Why do we require heating? The reasons for, and why do we use a water bath? I want you to look at these 3 points that you can see on the text above the screen. Reasons for using water bath is because heat की even distribution होती है. It prevents the test tube from bursting. China की सस्ती सी test tube है. And direct heating will cause the color of the test tube to turn black from underneath. And at the end of the day, it's the color changes that we're trying to observe. Okay, so अगर color change हम सही तरह observe नहीं कर रहे because of the black nature of the test tube, then that defies the entire purpose. And then this is the color change that you need to remember for reducing sugars. ठीक है, that is a very quick skim through that I've done. Of course, हमने करना इसी को है, तो I will jump on to the document that I have ready for you. Now, there can be instances today where we come across topics that you may have not done, like you can see a flower, but that's perfectly fine because the topic that we're doing is actually food tests. Okay, so you don't need to get much worried about कि ये तो हमने किया नहीं हुआ नहीं, हम food tests कर रहे हैं और हम food test को ही समझने की कोशिश करेंगे. So I want all of you to please pay attention. I want all of you to get a rough copy. इसको neat copy पे ना लिखें. If you don't have a rough copy at the moment. Haji, how is my voice now? Better? Just change my connection. Haji, better है कि lag कर रही है बच्चों को भी?
Yes, sir. Better.
Sir, can I do it on the copy?
Well, Asmina, I want you to make mistakes. So I don't want you to. I do it at the back side of your copy, not the main pages. Back side पे कर लें. ठीक है? Okay. Is everybody with me? Should we begin? Come on, I need good guesses.
Sir,
Sorry, again my voice is lagging. चलो मैं PT से भी आ जाता हूँ. क्या Wi-Fi मेरा जो है ना? हाँ जी, is everybody ready now? Yes. Okay, seems like nobody is ready. बच्चों, ready हैं? Yes तो कर दें. Okay, चल जी हम. Oh no, my. Okay, whatever the case is. चलें जी. So the first question says it's मैंने ऊपर year paper का year number भी दिया हुआ है. It's a slightly old paper, May 2012. This is question 3 of that paper. It says figure 3.1 shows a mature flower of lilium species. Okay. On figure 3.1, identify with a clear labeling line and its name. ये वाला portion अभी हम नहीं कर रहे because this is not food tests. So I just want you to ignore the A part. ठीक है? Just ignore the A part. अच्छा जी, और हम देखेंगे यहाँ पे B part. It says describe. Okay, so the first important thing that we need to understand is whenever you are looking at exam questions, बच्चों जो मैं आपको आज बातें बता रहा हूँ वो बहुत important है. Please understand. Describe is a glossary word that requires something from you when you're answering a question. Explain is a different glossary word that you require to answer the question in a different way. Okay? So whenever we are reading an exam style question, there are the first two things that I want you to understand is number one, the glossary word, which is described in this case. And number two are the marks. Okay. Our marks kitne hain? 5 marks. Okay. So now let me read the statement of the question. Describe how you might safely. Okay, that's a condition he's mentioning in the in case safely carry out food tests to show that these tissues contain reducing sugar. Okay, so there are tissues here, well, in the flour which contain reducing sugar. Now it's a 5-marker question. It's a descriptive question. और वो आपको कह रहा है कि आपने बताना है कि how safely you would carry out food tests to show that these tissues contain reducing sugar. Now, before I answer this question, Bacho, there is one thing that I would want you to understand very carefully. Okay, and that is, अगर मैं आपको एक example देता हूँ, you have, so let's say this is a room. Okay, so 2-dimensional room, for example. और मैं आपको कहता हूँ कि इसका आपने वॉल्यूम मेजर करना है, ठीक है? तो आप क्या करेंगे? Simply you will say that this is the height, this is the length, and this is the breadth, for example. ठीक है? और वॉल्यूम कैलकुलेट करने के लिए हम क्या करते हैं? Length into breadth into height, right? Is that the case? Yes or no?
Yes.
Yes.
अच्छा, अब this is not practical. अगर मैं आपको अभी भी कहूं कि for example the room that I'm sitting in at the moment, let's suppose is a square room, okay? And if I were to be asked to describe practically how will I measure the volume of this room, तो मैं क्या करूंगा? मैं सिर्फ ये कहूंगा कि length multiplied by breadth multiplied by height? No. What is the practical detail that you think I should be adding to my answer? Well, think about it. I mean, मैंने measure करना है, तो maybe I will need a measuring tape. Okay, that's a practical aspect to this. And then maybe ऊपर length measure करने के लिए मैं इतना लंबा नहीं हूँ कि मैं अपना हाथ से ऊपर छत तक करूँ. I would need a ladder, for example. Okay, and then maybe I would need a copy, and maybe I would need a calculator, and so on and so forth. You understand? So these are all the, and maybe I would need somebody to help me, a person, for example. So when you are doing ATP questions, you have to ensure that you change your frame of mind to practicality as much as you can. Okay, so if I were to write, for example, the same answer in an ATP exam, that I first have a measuring tape, that I measured the length first, and then when I have to measure the height of the room, I adjust the ladder at one side, I make sure that somebody is holding that so that I don't fall. That's part of the safety. और फिर मैं वो लेंथ लिख के साथ कॉपी पे नोट पे रख के, and then I do the calculation and then I write down the answer. I mean, at the end of the day, it boils down to this, but this only writing this thing lacks practical detail. You understand? Is everybody following?
Yes.
Okay. अब देखिए, now let's come back to this question. Now, I'm sure that you know कि किसी भी सैंपल पे अगर आपने टेस्ट करना है कि रिड्यूसिंग शुगर है, तो आप क्या करेंगे? बेनेटिक सोल्यूशन डालेंगे, उसको हीट करेंगे, और अगर कलर चेंज हो जाएगा तो reducing sugar is present. I mean, that's simple. Take the sample, add benetic solution, heat in a water bath, and then you have the results. But again, that lacks practicality. Okay, so let's come to the marking scheme. Look at this. How many points are given in the mark scheme? 1, 2, 3, 4, 5. और फिर देखें नीचे ये एग्जामिनर के कमेंट्स भी हैं, ये हम बाद में देखते हैं इंशाल्लाह. Let's see what the mark schemes have to say. So the first thing is tissue is prepared or sample is taken. आप उसको लेंगे, और सैंपल को प्रिपेयर करेंगे. Okay, that's the first mark. And then you add Benedict solution and then you heat it. Of course, यहाँ पे आप practical details जरूर mention करें कि you will do that with a water bath. ठीक है. मार्क स्कीम का ये मतलब नहीं होता कि बस जो मार्क स्कीम में लिखा हुआ है that's it, उससे हमने बाहर नहीं जाना. That's just a reference. Then the color change described. You have to be able to describe what's the color change. What's the color change बच्चों? क्या color change है बताएं? Blue to
Green,
Yellow, orange, red.
yellow, orange, red.
yellow, orange and red. Alhamdulillah. ठीक है, तो ये आपने color change याद रखना. And then use of water bath or any other. देखो use a safety feature का उसने अलग mark रखा हुआ है. Why? By the way, ये mark scheme के points के end पे जो आपको semicolon नजर आता है ना बच्चों ऐसे, जिस point के end पे ये होगा इसका मतलब है this is one marking point. यानी कि you will get one marking point, one mark if you have this sentence or something with respect to it. Okay. Now let's read what the examiner has to say. Listen to this very carefully. Okay. ये एग्जामिनर कमेंट्स हैं. एग्जामिनर ने सारे कैंडिडेट्स का ओवरऑल आंसर असेस करके अपने कमेंट्स दिए होते हैं और बताया होता है कि द गुड कैंडिडेट्स डिड दिस एंड द लेस गुड कैंडिडेट्स डिड दिस. इंशाल्लाह all of you are good candidates. Okay. It says many candidates had an obvious experience of the test for reducing sugar and were able to describe the test. Of course, alhamdulillah, we've done this also and state the expected results correctly. A few misinterpreted the question and incorrectly used the pollen grains as the tissue being tested. Pollen grains नहीं है, वो flower का हिस्सा हमने करना है. Of course, मैं उस detail में गया नहीं because the point is not to go into details of flowers, but the point is to go into details of how are we testing. The majority of candidates were able to name Benedict solution as the reagent used. See, so it's important कि आप reagent का नाम लें. Some candidates did not score full credit as they failed to suggest heating the Benedict solution during the test. You have to remember कि Benedict solution का जो test है, reducing sugar का जो test है, that is the only test that involves heat. So that point has to be mentioned. See, at the end of the day, it was a 5-marker question. Okay, so you have to write all of those points. Acha ji. The question asked for a description of how the test might be safely carried out. A number of candidates did not suggest the relevant safety feature, यानी कि use of water bath to heat the Benedict solution or use of tongs. Tongs कहते हैं वो एक इस तरह की scissor सी बनी होती है जिससे आप test tube को उठा लेते हैं. You don't touch it with your bare hands. Of course, when you're also using gloves, you're protecting your bare skin from touching the chemicals directly because chemicals they can cause irritation and so on and so forth. So that's basically how you would answer. अब देखिए answer इसका बहुत simple है. If your answer revolves around these 5 points, inshallah you will score all of the marks. ठीक है जी, is everybody good? समझ में आई? How to answer questions? Yes or no? चलिए जी आगे चलते हैं. Okay, now this is May June 2012. This is a lovely question. मैंने सारा question नहीं दिया हुआ, आप देखेंगे B part से शुरू हो रहा है because that is relevant for us. So let's look at this question together. All right, again, it has to do with food tests. Food tests were carried out on some cells containing storage granules as shown. ये जो storage granules हैं, ये storage granules they have starch. ठीक है, वो diagram मैंने include नहीं की हुई, लेकिन आप समझे कि there are some storage granules which have starch. Now,
Sir,
yes.
sir, could you try fixing your internet again?
अच्छा मेरा इंटरनेट ऑन कर रहा है, मैं 4G ऑन कर लेता हूँ. Okay, things should be good now. I'm on my data. हाँ जी, ठीक है जी आवाज.
Yes, sir.
चलें जी. Okay, so again, let me repeat. Food tests were carried out on some of the cells containing storage granules. storage granules याद रखें are starch storage granules. ठीक है? Okay, in figure 3.2 उसने simple figure दी हुई है जिसमें उसने इस तरह storage granules दिखाए हुए हैं और उसके अंदर starch होती है, simple as that. So don't get confused about कि diagram नहीं है. अच्छा जी, some cells were placed in water, shaken and then divided into two parts. Okay, thank you for telling me that I'm running out of time. Um, some cells were placed in water and shaken and then divided into two parts. ठीक है, ये देखें, उन्होंने क्या किया? Some cells they were shaken and then they were divided into two parts, A and B. इनके साथ क्या किया? A में they added Benedict solution, so A plus Benedict solution added, and then B plus Biuret solution added. Okay, now let's look at this particular situation. Starch के लिए बच्चों कौन सा टेस्ट यूज होता है? Iodine.
Iodine solution.
Iodine test. Okay, so if there's starch present, then of course, what are we doing with Benedict solution? What are we doing with reducing with Barrett solution? I mean, that's a question mark that we're posing. The examiner, you have to understand, the way I'm interacting with the text, it's very important that you develop this habit also. I know it may seem a bit difficult at the moment, but the more you interact with the text, the more it is conducive for you to make lesser mistakes. यहाँ पे वो कह रहा है, some cells were placed in a dry test tube. Now, this is dry, this was water, and then this यहाँ पे C में हम क्या add कर रहे हैं? This is dry, C plus iodine solutions. Now, that looks good. That looks good because starch is supposed to change the color of iodine, sorry, is supposed to change the color of the starch. चलिए आगे देखते हैं उसने क्या कहा है. State the color of the drops of liquid being added to each test tube. तीन liquids add किए हैं बच्चों, Benedict solution, Biuret solution और iodine solution. ठीक है? So state the color of drops of liquid being added to each test tube. Test tube A में Benedict solution add किया. What is the color of Benedict solution?
Blue.
Blue. What is the color of Biuret solution? What is being added to B? Test tube B. कौन से drops add हो रहे हैं बच्चों? ना मेरा बच्चा, Biuret is again blue. Okay, and then iodine. What is the color of iodine?
Brown.
Brown, orangish is acceptable. Let's look at the mark scheme. ये देखिए, blue, blue और yellow slash brown. Brown, orangish कर लें, ये थोड़ी पुरानी mark scheme है. It's usually brown, orange, brown and orange. Okay. Table 3.1 shows the results after the tests were completed. अच्छा जी, observation है जी, A के अंदर green solution बन गया है. Okay, that's interesting, isn't it? Let's zoom out a bit. Test tube A में cells हमने डाले थे, water भी डाला था, Benedict solution भी डाला था और color green आ गया है. That's interesting. What can we conclude for this observation? क्या ख्याल है बच्चों?
Sure,
What do you
there
think?
is like slight reducing sugars because like green is like the second color.
यानी हम ये कह सकते हैं reducing sugar present है, कह सकते हैं, because color change हो गया ना. देखिए, blue to green to yellow to क्या था जी आगे?
Orange and red.
Orange and
Sir,
red.
and then brick red.
Yes, red and brick red, one and the same thing. ठीक है, तो जरा सा भी कलर चेंज हो गया, अब ग्रीन आ रहा है, इसका मतलब अच्छा, अगर ग्रीन की बजाय ब्रिक रेड आ जाता, तो what do you think would have been our conclusion? We would still conclude that reducing sugar is present, isn't it? Yes or no?
Yes, sir.
But then what's the difference between green and red? What does that indicate?
Sir, green
So
is like very
ज़्यादा-
low concentration and red has high concentration, very high concentration.
Excellent, perfect. तो यानी कि मैं ये आपको सिर्फ as a side note बता रहा हूँ. Of course, this is not being asked in the question. So green is indicating lesser of a concentration of reducing sugar, while red is more. जी अच्छा यहाँ पे हम ना देख लेते हैं, मुझे थोड़ा सा सांस चढ़ गया. Let me end the meeting. Let's rejoin and then begin. Please re-enter the meeting.